Stanford University.
It's really an honor to be here with this group. I want to share some of our work, but I want to take a minute beforehand just to acknowledge the long history that's gone before us in making this institute a reality. And I want to tip the hat, of course, to the people who worked on this idea for so long. Lubert and Karl, the faculty planning committees, Rob and Li Chun and Gary Steinberg and Brian and that whole crew who worked on making this actually happen. And then everybody who came together at the administrative level, John Hennessy, John Etchemendy, Ann Arvin, and those folks who made it happen. And I think the spirit is really wonderful in this room. We saw that yesterday, and I think the future is very bright. So I want to share a lot of work that is really focused on this problem of how we can study intact systems, intact biological systems, but at high resolution without taking them apart. And we've used optical methods to do this in a variety of different ways. And one theme is that we've tried to do this in such a way that we're building toward keeping things intact, but also all the different tools might be mutually compatible so that we could get different data streams coming from the very same preparation, the very same animal, for example. And this is, I think, relevant. I'm in both bioengineering and psychiatry, and I think people from those disciplines appreciate this need, but I think all neuroscientists appreciate that as well. This human brain is, of course, a major challenge, but we're starting with animal models of various kinds, which have their own beauty and challenges as well. We use optical tools to control freely moving animals. We're using optical tools to look at structure and optical tools to look at fine patterns of activity, cellular resolution, and beyond in intact and behaving systems. And we're working toward trying to make sure that these are all mutually compatible. Now, the first thing I'll talk about is this thing in the lower right. This is a structural tool, and we call it Clarity. It's a way of keeping the brain intact while allowing high resolution exchange of information. And the way that works is some interesting chemistry at its core. It allows, ultimately, the very vigorous removal of lipids from the preparation. And that is important for allowing macromolecules to gain access for labeling purposes, but also in reducing greatly the scattering of light and achieving transparency. But you can't do that immediately. You have to first make sure that that process won't destroy your tissue. And so what we do before that is we build a three-dimensional network of a polymer, a hydrogel, that's linked covalently to the native amine-containing biomolecules. And there's a hydrogel monomer, in this case acrylamide, which after this reaction and the loss of the water leads, the monomer is then covalently linked to native biomolecules, nucleic acids, and proteins that have amines, but not lipids, which tend to lack those groups. And this is then polymerized into a gel, which has a very dense structure, nanoporous form, and it keeps the proteins in place even at the ultrastructural level. Then lipids can be removed. We initially did this, and still do sometimes, with an electrophoretic method, which is fast. If you can tolerate waiting a week or two for your adult mammalian brain to be completely cleared, you can actually do it without that electrophoretic step at all, as we've reported just a couple months ago. This is work from Raju Tomer, Li Ye, and Brian Shui in the laboratory. And this we call passive clarity. It's a lot easier. It just takes a little longer. And the end result is the same, is that you still have to build the hydrogel in place beforehand, but you end up with a transparent brain. Now, what also sets us apart is the lipids are gone, and so you can then use antibodies and nucleic acid probes to molecularly interrogate the tissue. But there was another problem which we had to address, and that had to do with this sort of new opportunity, creating this new problem, which is you've got an intact structure that you want to collect information from at high resolution. But typically with confocal or two-photon, you're moving a point of light, you're illuminating your whole tissue, and it takes a very long time at high resolution to get your information out. That creates a bleaching problem in addition to just taking a long time. and you've occupied you know, your expensive machine for a long period of time just to acquire this information. And there's a 100-year-old idea, light sheet microscopy, which is now, I would say, really emerging into widespread use in neuroscience and for activity imaging. And Raju in the lab has been instrumental in leading the development of what we call clarity optimized light sheet microscopy, a way of using its very special properties, but with fundamental innovations on top of that in order to allow rapid imaging of the tissue. And the way that works is you scan a sheet of light, two-dimensional sheet of light, instead of a zero-dimensional point effectively, and so it's much faster, but also you're only illuminating where you're also imaging, and that you know, really addresses the bleaching problem very well. Now, there were some challenges, and it's described in the publication. I won't go into in great detail, but light sheet historically has had some challenges with the resolution, particularly in Z. Good for sort of cell body level resolution, you know, 15 microns or so, but for neuroscientists, that's not quite enough. We really want to see the wiring if we can. And so this sort of next generation light sheet methodology allows us to get to that level in an intact adult mammalian brain that's transparent. This is a clarified, as we say, mouse brain, a little cuvette in refractive index matching solution. There are objectives now. You know, when we started a year ago, there were no clarity optimized objectives, of course, but now there are. And you can buy these from Olympus or Leica. This is an Olympus one that has beautiful properties. We've got, some of these have 8 millimeter working distance and very high NA, and we can actually do really fast and high resolution investigation of the tissue. And here's an example of that. This is a confocal acquired section. It took a whole day to acquire this, though, at high resolution. Pretty, you know, you can see cell body and process level resolution even deep, but it took a long time to acquire. With the light sheet method, this took 2 minutes to acquire, but same comparable level of resolution even deep, and allows us to really take this to the next level. This is an intact brain, adult mouse brain of the thigh 1 YFP type, and so the label here is in long-range projection neurons. And it's imaged intact. There's no staining in this case. I'll show you whole brain staining in a moment. But the fluorophore is a native fluorophore that is preserved in this whole process as well, which is not a foregone conclusion either. A lot of prior methods for achieving transparency tended to greatly bleach endogenous fluorophores, in addition to not allowing the lipid removal for subsequent staining. And we get the resolution that we want, even deep in tissue, and we can do whole brain staining and then whole brain imaging. For here, a couple examples, tyrosine hydroxylase, and also for parvalbumin, a very interesting kind of interneuron in the brain. Now, that's published. I want to show you some unpublished work. What we've found is, you know, that we can adapt these very same methods and the same preparation to look at activity, and this allows us to really bring things together. Now, a lot of groups have been really beginning with work from Misha Ahrens and his colleagues at Genelia have been engaged in using light sheet in conjunction with genetically encoded calcium indicators in order to image activity in zebrafish. And that's what we're also doing here. This is actually, though, what's interesting is this is a somewhat broader field of view, higher resolution, and somewhat faster, but same kind of thing in principle. But what's unusual about this is that this is being carried out on the very same light sheet scope that we can also then acquire the detailed wiring from in that same animal. So we can, you know, a lot of you might know that there's been a vigorous debate in the neuroscience community. How useful is this sort of data set? We've got all these beautiful flashing lights, and you look at them and you think that's just amazing. But it's kind of hard to go beyond that step next. I mean, there are things you can do. You can assess the statistical properties and the dynamics, but there's not a lot of extremely deep insights into how circuits work unless you know more about these flashing lights. And if any of you saw on September 30th, the BRAIN initiative meeting at the White House, it was live webcast, but Corey Bargman actually said that more or less explicitly, and I think that's widely appreciated in the community. And so what we can do is we can take these very same fish, use the GCAMP label as our structural label, and even the same individual, we can then having seen those patterns of activity, even having presented behavioral stimuli to the fish, we can then fly in and see how those neurons are connected and who they are. Now, this is very downsampled. These are huge data sets, which gets into another interesting question. You're not appreciating the full resolution just so we can play this movie on my laptop, but we're getting many terabytes of data even from a single round of experiments, and we're getting to petabytes on the sort of weekly or monthly scale, and it's actually a real interesting computational challenge, and it's an exciting thing to be here. I think at Stanford, where we have perhaps the premier computer science group in the world, and bringing those sorts of talents to bear is going to be absolutely crucial for the future of neuroscience. Now, that structural aspect and function linked to structure, of course, is very important. But what we've also worked on is trying to understand which of those activity patterns are important, causally important. And for that, we use tools like optogenetics. I won't go into a great amount of detail, but we have a couple primers published in Cell and Neuron a couple years back that have some of the basics if any of you are interested. But the way these proteins work, there are both channels and pumps. They're borrowed from microbial organisms that do things very simply and efficiently. They use a single gene, a single protein, to achieve both light detection and ion flow. And the pumps and the channels work in very different ways. The channels actually open a pore and allow cations, sodium, potassium, calcium, and protons to go through. The pumps are set up quite differently. They do move ions, but they only move a single ion per photon. And it's in this discontinuous fashion, so there's not an actual input resistance change in the membrane of the cell. But there are many of these opsins found across all the major kingdoms of life. We and many others have been using genomic strategies to find new ones with new and interesting properties. But an interesting theme had been that all of the channels that we found were cation channels, and under physiological conditions, that means they're excitatory. And that's great. The pumps were all inhibitory, whether chloride or proton pumps. But one thing we and many people wanted was to find or create an inhibitory channel, which would be much more efficient, move many more ions per photon, and also create input resistance changes and allow other kinds of interesting manipulations as well, having to do with the kinetics that I'll show you. But for whatever reason, despite all the searching that many labs around the world were doing, nobody had found a inhibitory channel. So we engaged in an effort to try to understand these proteins more deeply with the hope that we could achieve a engineering strategy to create an inhibitory channel. And so we teamed up with a crystallographer, Osamu Nareki, at the University of Tokyo, and this collaboration took many years, creation of many different chimeras, trying to find the ones that expressed at high enough levels, and that still conducted current and allowed eventually the crystal structure of channelrhodopsin to be determined. And that allowed us to then see the pore. And it is a dimer. It's a beautiful structure of two 7-transmembrane proteins that each have an all-trans retinal embedded within them that is actually the chromophore. And there are pores that each monomer has. And we could see that at high resolution. And here, a tip of the hat not only to Feng Zhang and Ofer Yuzar, but also Shara Ramakrishnan, who made all these constructs in the lab and allowed this to happen. Now, having this high-resolution information on the pore was, of course, extremely interesting. We could see the positioning of all the side chains and the charge and polar residues that form the pore. It's a closed-state structure. We're working on an open state. Don't have that yet, but we can still see the pore. And we could work on strategies to create an inhibitory channel. Now, given the ion balances that are typically found in physiological systems, either creating a potassium or a chloride channel might be effective. We went down both routes at the same time. We're able to create some potassium selectivity, not enough to actually create an inhibitory channel. But the chloride approach actually worked. And it was really satisfying. If I'm, more, I think, a patch clamper than anything else, and this was the assay we used. It's not the high throughput assay, but it was really nice for us to be doing this, and just thinking back on how this very simple patch clamp apparatus with the little suction that lets the gigaseal be achieved 35 years later, it's still, I think, the gold standard. As exciting as optical techniques are, if you do the patch clamp, you really know what's going on with your neuron. You know your neuron very well at that point. And that was incredibly important for this set of experiments, because we wanted to make sure that the neurons were healthy, look at their resting potential, looking at their input resistance, membrane dynamics, and understand the reversible potential of the opsins we were creating. And so we expressed these opsins in cultured neurons, which Charu prepares. And these are very beautiful cells. They are difficult to patch clamp, as you can appreciate, but we at least get the information that we need out. And Andre Berendt and Sue Lee carried out this very extensive series of mutations, both alone and in combination, throughout the pore and in the vestibule of the channel, looking at those that had improved chloride conductance, which didn't exist at all. So first having it and then making it more and more robust. The mutant switch had good photocurrent and had some effect on chloride conductance, were then assembled into 4-fold and 5-fold groups, and then into a full newly upholstered channel, which took the electrostatics from largely negative inside the channel to largely positive, as indicated by the red to blue shift. And this actually created an inhibitory channel. It expressed well in neurons. We called it IC1C2. It was published back-to-back with a very nice paper from Peter Hagemann's group, arriving at a similar result through different means, starting from photocycle considerations. Both groups achieved a chloride conducting channelrhodopsin. And here we're driving spikes either with current injection, steady or pulsed, and we can inhibit spikes with blue light. And one interesting aspect is with inhibition, very often you want to keep it on. Although there are cases where pulsed inhibition is important, it's not typically what people want when they're doing inhibition. They typically want stable inhibition for a period of time. And one reason that the channels were so interesting to create an inhibitory version of this is that we knew from several years before that we were able to make bistable channelrhodopsins. With just a single pulse of light, you could flip them into a stably activated state. And this we did back in 2008. Native channelrhodopsins turn off very quickly with about a 10 millisecond time constant after you turn off the light. And these mutations actually stayed on. You could actually still turn them off with a different color of light, and this was due to details of the photocycle. You can shortcut back through to the dark state with a different color photon. But what was interesting about these, because the opsins don't really turn off, the cell that is expressing many of these opsins becomes like a photon integrator, and you can get very weak light pulses for very long, and you can still get to the maximal photocurrent that the cell can achieve. And it's an orders of magnitude effect. You can make cells that are vastly more sensitive to light. But also you get this bistability, so you don't have to keep putting light into your tissue. This is how the excitatory step function opsins, as we call them, worked. You can deliver 3 identical stimuli to a neuron. But if you've flipped the opsins that it's expressing into, and these are electrical stimuli played in through an electrode, but if you can flip it into a excitable state with a modest current through its step function opsins with a pulse of blue light, you can spiking is elicited, and then you can take it out with a pulse of green light. Same neuron, same continuous trace. Without that boost, the cell is not spiking, and then you can flip it back into the excitable state. It's a nice way of also maintaining the cell's spiking, having a relationship to its input rather than driving spikes when you want them to happen. But this bistability, then, we thought, well, let's take those same mutations and apply them to our inhibitory chloride channel, and let's see if we can achieve switchable inhibition. And indeed, we can. We call these SwiChRs for step waveform inhibitory channelrhodopsins. And these are really interesting for their bistability, their increased light sensitivity. We found they traffic well. We can inject them into the basolateral amygdala, and then they can show up in downstream structures, and that's very important for a kind of targeting called projection targeting. And actually, Scott Delp, our colleague and collaborator, has wonderful results using these SwiChRs in a pain model in freely moving animals. Now, I'm not going to talk about that, but I will talk about some applications, recent applications of these sorts of methods, optogenetic methods and activity detection methods in intact animals and behaving animals. The projection targeting concept I mentioned is very useful. It's very versatile. You don't need to have a lot of genetic information about the animal to make this work. You can transduce a deep brain structure with an opsin, and if you have a promoter or a recombinase driver animal, you can make that selective for a subset of cells. And you can deliver your fiber optic, which gives you this spatial specificity as well, and you can recruit a very local population of cells. Or you can deliver your fiber optic downstream, and you can get a population of cells that's defined by having a particular connectivity, and you can do either excitation or inhibition. And this, it's an interesting historical tidbit. The first person to do genome editing, Feng Zhang, was also the person who designed this system. So this is clearly a guy who knows how to build things that work. He's pretty impressive fellow, and he worked together with Alex Aravanese, an electrical engineer in the lab, to design this interface. And the number of things they wanted to do to make this work, they wanted to, knowing that light, by itself, of course, is a nice method for delivering information into tissue, but they wanted to keep the heat generation separate. They know that heat not only kills cells, but even at very small levels, tenths of a degree, it can bias excitability of cells, which they didn't want to have happening. So they worked very hard to make sure the laser diode stayed separate and the fiber optic was just carrying light in and not heat. And this has turned out to be very versatile. You can do, building on the knowledge, of course, that animals can, rodents, rats, and mice carry out beautifully complex behaviors while recording happens. For example, we just saw that in the Nobel Prize that was awarded earlier this week to O'Keefe and the Mosers for electrical recording from freely moving rodents. Well-validated behavioral tests in this sort of situation, and a lot of wonderful work. And so this is actually something that a literature we could build on and make sure we were doing well-validated tests. The fiber optics are very light and thin and allow complex behaviors, running, jumping, swimming, social behaviors, and so on. And here's an example of that. This is what's called an elevated plus maze. It's an anxiety-related measure. And there's the closed arms, which have these elevated walls that the animal prefers to be in most of the time. There's the exposed or open arms, which the animal tends to avoid. But one thing we found in 2011 first was that there was a projection from the basolateral amygdala to the central nucleus of the amygdala, BLA to CEA. And if you drive that projection optogenetically, as is happening now, for the first time, the animal will venture out into the open arms and include those in its exploration of the system. And so this is the kind of information you can get using this projection targeting method. And the proof is in the pudding now. Thousands of investigators are using this tool, and there are hundreds of published findings using this sort of method, different projection origins, projection targets, finding the effect of influencing circuit dynamics along certain projections or cell types. And here, just a review summarizing the anxiety and the depression-related tasks. And that is, of course, of great value. We've just begun to scratch the surface, but there's a problem, which is even though this gives us access to the cell type that's of interest and the projection type that's of interest, and we can bias the dynamics, and that's going to continue to be useful, we would actually like to get to the single cell level if we can, play in multiple single cell patterns of activity that might be asynchronous. And this is something that is, of course, a challenge. And we would like those to not be completely randomly selected. We'd like those to be guided somewhat by what we are seeing. And if we see a pattern of activity, we might like to provide that same pattern of activity and see how that affects behavior. And we're taking steps toward that bit by bit. It's a big challenge, but the nice thing about the fiber optic interface is you can collect information back from the very same preparation. And this is in the same device. And this is a very important opportunity for mammals, which, of course, are non-stereotyped in their detailed wiring. And it would be very nice to see for a particular animal what the particular pattern of activity is, and then to provide that back and see what happens. And so this question of observing dynamics is important. And so we've been working on this and using the fiber to collect information back at the same time. or in parallel preparations. And this set of work was a collaboration among several folks in the lab. The photometry technology itself developed by Logan Groznik and Isaac Kalvar in the laboratory, and Lisa Gneiden and Joel Finkelstein ran very interesting social behavior tasks in animals that had this device in place. And we use genetically encoded calcium indicators to detect the patterns of activity, in this case GCAM 5.0. It's even better with the more recent GCAMs that you can use now. And we deliver these genetically encoded calcium indicators, which several groups in Japan and at Janelia have created, into targeted sets of neurons. We can use Cre driver animals, for example, that express Cre recombinase only in a cell type, like the tyrosine hydroxylase cell type. Then the fiber gives us intersectional specificity because we can go into just one brain region, in this case the ventral tegmental area, avoiding other TH cells that might be in the brain. And we can look at the local pattern of activity in the dopamine neurons that are in the VTA. And it sounds good in principle and practice. It's a small signal, and you've got a freely moving animal, a lot of artifacts that are possibly going to be present, fiber bending and so on. And so there, it's what's described are a number of tricks, including a lock-in amplifier that helps make the signal cleaner and more robust and helps exclude sources of noise. But you can actually end up doing this in a brightly lit room if you just express yellow fluorescent protein by this means, and you do a task, in this case sucrose consumption, and you use a lickometer, so every lick that the animal makes on this little spout to get a highly appetitive sucrose-containing solution is picked up, as indicated by the red bars. You don't see an activity-dependent signal. The case, though, where you have a genetically encoded calcium indicator present, you then see something very different. You see this habituating but time-locked pattern of activity in the dopamine cells in the ventral tegmental area as the animal behaves. And so this is it's not an image. It's a time-varying scalar, which is reporting on the activity of your population of cells where you've introduced your fiber. And what's particularly exciting about this method is we can then do the projection-specific version of this. We can move our fiber to a downstream structure, in this case the nucleus accumbens, and there's a robust projection from the ventral tegmental area to the nucleus accumbens, which has been hypothesized to be involved in various reward and motivational related behaviors. But nobody had been able to observe the time-varying traffic along this projection in real time during behavior. And there were interesting questions. If one could develop a versatile technique to allow this sort of thing to happen, it potentially could be applied to many different situations, observing the traffic along particular projections during behavior. And this actually works, so we can, we knew that there were interesting differences that optogenetics had taught us in terms of the causal effect of activity along this projection. But we didn't know how the animal was normally using the projection. So this gets back to the theme of seeing how the cells are normally active. But if we go in and do the fiber photometry, we actually see a very much corresponding result, which is that a novel object exploration does not give rise to a very robust signal along this projection, whereas the social interaction does. And this was a difference that was not seen at the cell body level, so it actually required our ability to see this projection targeting. And I'll just show a couple movies to make this point. Now we're looking at time-varying activity arising from the ventral tegmental area tyrosine hydroxylase cells and hitting the nucleus accumbens, and effectively the time-varying traffic along this link. There are other ways to approach some of these other interesting parameters, which I won't talk about yet. Top is social, bottom is novel object. This oscillating red bar is that signal, and you can see it's kind of interesting to watch over time as the animals engage and then separate and re-engage. There are little blips in the novel object case, but the animal doesn't seem that excited about the golf ball. So it's so this is but of course even this is not as good as we'd like. We'd like to get to the single cell level, and so we're working on that. It had been historically the case that the opsins were not very good at being driven with 2-photon illumination, which was crucial to getting to the single cell level intact in the intact mammalian brain due to scattering. But one of our chimeric opsins engineered in a few different ways that is called C1V1, actually works for that. And you can do simple raster scanning, 2-photon illumination, even in an awake mouse. This is a paper led by Rohit Prakash in the lab. It came out a couple years ago. Collaborators were Rafa Yusta and Mark Schnitzer as well. And you can patch a particular cell in cortex here filled with a patch pipette filled with a red dye, a loose patch recording. And you can scan a plane just above the cell or just in the middle of the cell or just below the cell, but only when you're scanning in the plane of the cell do you actually see spiking. So single cell resolution, that's one cell. Now, of course, that's not good enough. We'd like to control multiple single cells, and of course, we'd like that to be the cells that perhaps even that we've observed a particular pattern during behavior. And so this is sort of the next step. We're scratching the surface of this in many ways. I'll show you one example of many different sets of work that are being done in the lab along this front. This is work led by Jim Marshall along with Will Allen, Sam Yang, Rohit Prakash, and others, but there are others in the lab doing this sort of thing, including Tina Kim and Logan Grosnick and others. Here we have C1V1 expressed for excitation and GCAMP6 for imaging. It's all done with 2-photon and an awake mouse, looking at layer 2-3 pyramidal neurons in mouse visual cortex. And you can do a couple different interesting things. You can play in sequences of activity, hop from cell to cell, defining regions of interest, and play in blips and elicit patterns of activity in particular sequences. That's done sequentially, but you can also use a spatial light modulator, which is basically a liquid crystal-based holographic device, and you can do simultaneous multiple single cell imaging and stimulation. So you can observe natural patterns of activity. You can see that going on, I think. But here, this is when the light stimulation is coming on, and you can see for these cells, and this is a quantification of that, an association of the increased signal when those particular cells are being stimulated. So we are now at the stage where we can do multiple single cells, even in volumes in awake animals. So, you know, many things left to do, many directions left to go, but our goal, I think, and a goal that many share is to see if we can take these patterns of flashing lights and link those up to anatomy, wiring, molecular phenotype, and, of course, you know, the causal significance in behavior. I want to say a final couple things, just for a couple minutes, on sort of sharing and translation of techniques. And this is a thing that it has been wonderful to be at Stanford for this reason. We have a very open environment. I think that's something that everybody here appreciates and has been the case for a very long time. But that spirit is enabled by I think, very thoughtful consideration. And I'll tell you a little bit of a story. Early on in the optogenetics days, you know, a lot of people wanted to learn it, but it took about 4 years for the technique to really take off. Partly we had to solve a lot of problems, technical issues having to do with expression and designing the fiber optic interface and so on. But between 2005 and 2009, a lot of those problems still had to be solved, and there were very few papers published in the field. That all changed right around 2009, but there was a period of time where a lot of people had trouble getting it to work. Pretty stressful for me, you know. I got irritated emails from people saying this clone doesn't work, and, you know, we tried to help as best we could. A lot of these, you know, folks were not, you know, these were not natural tools to a lot of the people who would naturally want to do this sort of thing, fiber optics and viruses and so on, but it was pretty stressful, so we tried to bring people in to train them, and this actually worked a little bit, but we'd pack them into the lab. Here's what we were doing early on. Here's Anne Graybiel. She came out with her whole lab to learn some of these methods from MIT, and this is right in our lab in the Clark Center. And I was an assistant professor then at the time, so I didn't have you know, a lot of space and so on, but there was a really great conversation. You know, Bill Newsom and Karl sort of saw this situation, and you know, we chatted a little bit about it, and the idea of creating a optogenetics training space came out of that, and it's turned out really well. You know, and it's that kind of you know, spirit, I think, that's continuing, and you're seeing that in this room and in the long-term plans that SNI has. But we've been able to take this into a much more relaxed setting and extending this sort of concept. We're training people in clarity as well as photometry here, and people learning hands-on how to build some of the devices. Here's Tom Davidson in the lab showing people how to deal with a lock-in amplifier approach. And so this actually is something that it's good for many reasons. Takes some stress off of me. I don't need more stress, but also really helps people learn the technique. And one consequence of this is that, you know, the clarity method, it's only been a year, but already we're seeing papers from other groups already being published using the method, which is pretty interesting. And the clarity optimized light sheet microscopy is also being now adopted. We have websites, the clarityresourcecenter.com, with clarityresourcecenter.com, which has a lot of the tools and techniques and papers, and you can go and look at them, and general clarity resources as well. Some of the papers, here's an interesting one. I don't know this group, but this is looking at Alzheimer's brain. This came out in ACTA just a couple months ago, using clarity to look at plaques and tangles and axons in 3D. And they actually looked at a few different methods and actually compared them. They saw pretty good results with clarity, but the exciting thing was that you can actually, because it's human tissue, there's not a native fluorophore, and you can go ahead and stain for these important pathological markers and understand them. Here's another paper comparing gray and white matter using clarity, looking at mouse models of multiple sclerosis and looking in detail at gray matter atrophy. And a spinal cord study looking at the arrangement of pain fibers and using the intact volumetric approach to actually see which fibers are crossing the midline, and interesting palisading of some of these interneurons that are present in the spinal cord, which you couldn't appreciate without the 3D approach. So this is, trying to share these techniques, I think, is a big challenge, but it's something that we're now getting better and better at, and I think it's something that is going to be part of the sort of the DNA of the work we do, and I think of the Institute going forward. So I want to thank these amazing folks who did all the work in the lab. The aspects that I particularly focused on. Raju Tomar led the development of the light sheet microscopy. We're amazingly fortunate to have. He actually is really a world expert in light sheet microscopy, and given the really, I would say, exploding importance of light sheet and neuroscience methods, we're very lucky to have this talented scientist here at Stanford. And his collaborators on that work, particularly also in developing the passive clarity method, Li Ye and Brian Shui, Kwang-Hun Chung and Viviana Gradnaro, now at MIT and Caltech, led the development of that method in the laboratory. Kwang-Hun Chung is doing great work at MIT, as is Viviana at Caltech. She just had a cell paper on a next generation clarity method. I mentioned the photometry work, Logan Groznik and Isaac Kalvar, and the social behavior work, Lisa Gneiden and Joel Finkelstein. And turning channelrhodopsin into a chloride channel. This was work led by Andre Berendt in the lab, and also Sue Lee as well, side by side with him, doing all that patch clamping. And I mentioned the approach of this, using the holographic methods and achieving the all optical play in and read out of activity work that's led by Jim Marshall, Will Allen, Sam Yang, and building on work from Rohit Prakash earlier in the lab. And the fish work with integrating the structure and the activity, Raju Tomar and Matt Lovett-Barron in the lab led that work. Also want to thank all our many collaborators. I've mentioned many of them over the course of the talk, and our great staff. Those are incredibly important folks as well. I want to thank a lot of people in this room helped get us funding for some of these crazy projects, including Tom Insel. A lot of this work was, pretty high risk, I would say. If you were to describe, go back 10 years and say this is what we're going to do, hard to get that through a typical study section, I think. And so I really want to thank everybody who had the vision to support what we were setting out to do. Maybe not easy to appreciate, and that includes many people here at Stanford as well, who I've mentioned. I want to thank all our students and postdocs, who are all students and postdocs in my laboratory who are listed here, and everybody for their time, and looking forward to the rest of the day. Thanks.
For more, please visit us at stanford.edu.